It's my pleasure to introduce Dr. Jordan Meier. Jordan is currently a senior investigator or fully tenured PI at NCI in the chemical biology laboratory. Is that right? Yeah.
Yep.
So he got started off Midwest adjacent, I guess, in Creighton, where he did his undergraduate work, and then moved to UCSD, where he did his PhD with Michael Burkhart, where he got his real start in biochemistry and proteomics. Afterwards, he did his postdoc with Peter Durbin in his lab, working on high throughput sequencing methods and analyzing small molecule interactions with DNA. And then in 2013, he moved to start his own group at NCI, where his lab has been doing some really great work in using chemical approaches to investigate epigenetic signaling pathways, and with a particular focus on the acetylation of histones, which today he's going to tell us how histone acetyltransferase inhibitors are, or at least he thinks they're hitting hitting the big leagues, and that we're going to be using them quite a bit in our future research. So, Jordan, the floor is yours. Take it away.
Awesome. All right. Well, thanks, Jackson, for that kind introduction. I do have to correct you. I think Omaha is not Midwest adjacent. That's about as Midwestern as it gets. I think it actually has the most American accent. It's been judged in all of the U.S. Like, I don't know how they judge that exactly, but anyway.
Yeah, I'm from Illinois, so I have a much more Illinois-centric view of the Midwest. So
I'm from Iowa originally. So anyway. But anyway, so that aside, yeah, it's wonderful to speak at this seminar series. I remember it, finally, during 2020, I think is maybe when it started, but during COVID, kind of being a nice way to kind of connect intellectually with other people. And so I'm glad you all are keeping it going, and yeah, pleasure to present. So yeah, like Jackson said, my name is Jordan Meier. I'm a senior investigator at the NCI. Today, I'm going to tell you about work our lab has done developing chemical probes of histone acetyltransferase enzymes. Okay, so I don't need to give a lot of introduction for this audience, but histone acetyltransferases, or HAT enzymes, catalyze this reversible modification of the epsilon amine group of protein lysine residues with this reactive thioester of acetyl-CoA. So this results in a relatively small chemical modification, but can actually have a profound effect on many biological processes relevant to cancer, most notably gene expression. So shown here, just as an example, is the crystal structure of the nucleosome. The core component of chromatin consists of 146 base pairs wrapped around 2 pairs each of the 4 core histones. Now, histones, of course, are highly basic proteins, particularly their N-terminal tails, these stringy bits protruding here. So these positively charged tails are really important because they help electrostatically interact with that negative phosphodiester backbone, holding chromatin in a silent or inactive state. Now, in this context, histone acetylation is actually really useful to stimulating gene expression in a couple of different ways. First, acetylation neutralizes the positive charge of lysine, thereby reducing its interaction with DNA backbone. This allows chromatin to relax and allows transcription factors to come in, bind, and activate gene expression. Second, histone acetylation provides a binding site for bromodomains. These are small protein structural motifs found in many different classes of transcriptional activators that interact very strongly with acetylated lysine residues. It's really through recruitment of bromodomains that histone acetylation provides a mechanism for nucleating protein-protein interaction mediated signaling cascades that really ultimately culminate on mobilization of RNA polymerase II and stimulation of transcription. So really getting at the importance of this process for malignancy, cancer genome sequencing efforts have found many genes involved in the regulation of histone acetylation, in particular HATs, are common targets of oncogenic fusions, focal amplifications, and inactivating mutations, and they can really drive transformation and proliferation across a wide range of cellular contexts. So if histone acetylation were actually important to cancer, one might hypothesize that drugging it could provide therapeutic benefit. And indeed, really over the past 20 years or so, targeted acetylation has become a validated paradigm in oncology. This is demonstrated best, I think, by the FDA approval of 4 HDAC inhibitors for the treatment of cancer, as well as the widespread preclinical and clinical exploration of bromodomain inhibitors, agents that target this acetylation-dependent protein-protein interaction, one of which is currently in phase 3 clinical trials. So really missing from this arsenal have been histone acetyltransferase inhibitors, and actually no FDA-approved HAT inhibitor yet exists or has even reached phase 2 clinical trials. Okay, so for today's talk, I'm going to attempt to explain why developing HAT inhibitors has been such a challenge and also highlight some recent exciting progress in this area by really going over 3 topics. First, I'm going to introduce the rationale for targeting HATs in cancer and explain how novel assays can be used to separate authentic HAT inhibitors from artifacts. Second, I'll describe the recent discovery of drug-like HAT inhibitors and highlight some work from my own group, benchmarking the potency and mechanism of these reagents. And finally, I'll tell you how we're using chemical biology to help us build HAT inhibitors with new capabilities, including conditional release as well as protein degradation. And now that HAT inhibitors have arrived, so to speak, what new applications are in play. Okay, so we'll get started today by talking briefly about how new assays can help separate HAT inhibitors from artifacts. So the human genome encodes about 30 genes with known or putative acetyltransferase activity. Today, we're really going to focus on the 2 most well-characterized of these enzymes, EP300 and CREB-BP. So EP300 and CREB-BP, which are also known as P300 and CBP, that's how I'll be referring them to them throughout this talk, are 2 highly homologous HAT paralogs. This schematic shows our domain architecture. What I hope you can appreciate is that these are humongous proteins, about 300 kD in size, that house multiple domains, many of which interact with transcription factors. The histone acetyltransferase domain of P300 and CBP, that activity, which maps to this domain here in green, was actually discovered by Nakatani here at the NIH back in 1996, as well as independently by the Kozaridis group in the UK. So human cells contain thousands of sites of acetylation, and proteomic studies have actually found that P300 and CBP function as master regulators of a wide swath of these sites. However, their most well-characterized substrates are lysines 18 and 27 of histone H3, which I'll just refer to as H3K18 and K27. Now, due to their role as transcriptional coactivators, P300 and CBP have long been of interest as drug targets, and I'll just give 2 examples where this could be useful for cancer treatment. First, P300 and CBP are crucial coactivators of nuclear receptors, such as estrogen receptor and androgen receptor. These are transcription factors that, of course, play a key role in promoting anabolic gene expression programs in breast and prostate cancer, respectively. Now, it's been hypothesized that disrupting P300-dependent acetylation may limit the ability of these nuclear receptors to drive transcriptional signaling, and in the case of prostate cancer, perhaps even be effective in androgen-independent tumors, which have become resistant to drugs that are traditionally used to treat prostate cancer that target the AR ligand binding site. Second, as I mentioned earlier, P300 and CBP are mutated across many cancers. Now, this suggests they act as tumor suppressors. However, while loss of one is common, it's actually very rare that both paralogs are mutated. The reason for this was demonstrated by Ogawa and coworkers in 2016, who showed that tumor cell lines in which CBP is lost actually become highly dependent on P300. Likewise, knockout of P300 increases the dependency of cell lines on CBP. Now, we know from the work of researchers like Bill Kalin and others that this constitutes a synthetic lethal interaction, and it's hypothesized that this may create a therapeutic window for targeting P300 or CBP and the many different types of cancers where one or the other paralog has been lost. Now, while these examples really offer compelling rationale for targeting P300 and CBP, for a long time, the question has been how. Here, I think it's really useful to compare HAT active sites to those of the highly druggable protein kinase family. In crystal structures of protein kinases, such as ABL, here on the right, what we see is that kinases often engulf the adenine moiety of ATP. This really makes them ripe for inhibition by the myriad of flat aromatic molecules that are very well represented in our conventional high-throughput screening decks. In contrast, if you look at HATs, you find that they also bind to an adenine cofactor, acetyl-CoA. However, they interact most strongly with the 3' phosphate, as well as the phosphopanthine moiety, and actually rather minimally with the adenine, suggesting they may constitute a more challenging target for molecular recognition by small molecules. So in 2013, when our group started, the molecules shown here were really the state of the art for HAT inhibitors. Excuse me. There was lyse-CoA here at the top, a bisubstrate inhibitor that binds P300 very potently, but which is not cell permeable due to its high charge. There are also several low-potency putative small molecule P300 inhibitors. Of note, most of these molecules contain electrophiles or redox-active catechol, and at the time, their selectivity was really unknown. So our group's approach to help solve this problem was to develop new assays to study HAT inhibitors. Our thinking was that if this is, really a difficult class of epigenetic regulators to drug, then new assays could be critical in helping us characterize the known inhibitors, and if necessary, potentially also discover new ones. Now, in work I'm not going to cover in detail today, our group has actually developed and applied a number of different approaches to characterize HAT inhibitors. These include microfluidic mobility shift assays that can be used for direct biochemical monitoring of the HAT reaction, chemical proteomic probes that can be used to study the selectivity of small molecules that target HAT domains, as well as assays for monitoring cellular target engagement, for example, using a DCAS9 P300 dependent transcriptional reporter. Now, one of the most surprising outcomes of these efforts actually came in 2017 when we teamed up with a large collaborative group that was led by Jamie Dahl and Mike Walters and coworkers at the University of Minnesota, who used alarm NMR as well as other approaches such as mass spec to discover that a large number of the published HAT inhibitors were actually aggregators or electrophiles. Now, the reason this is a problem is because molecules with these properties will often show up as inhibitors in really any biochemical or cell-based screen, similar to if you added bleach or iodoacetamide to your assay. This discovery led to a meta-analysis of all HAT inhibitors that have been published in the literature to date. And what I found was that the vast majority of them, published before 2017, actually lack some aspect of critical characterization necessary to understanding their biochemical or cellular specificity, for example, such as assessments of reversibility or target engagement. Now, some of you may be asking why this matters. Can molecules that are, electrophiles also become drugs? And I would answer that absolutely. Of course they can, yes. However, if you have a molecule that's very nonspecific, it's not going to be useful for asking biological or therapeutic questions about histone acetyltransferase activity, and that's exactly what we want to do. I'll also just pause for a moment to point out that, poorly characterized molecules that masquerade as specific enzyme inhibitors can actually cause a lot of harm. I won't go through all these examples, but one that this audience is probably familiar with is resveratrol. This is a small molecule natural product that was initially characterized as a SIRT1 activator that was later found to be a biochemical artifact. GSK acquired a biotech, Certris, founded around the science for about $1 billion. This effort produced no drugs and is often credited as being responsible for a site closure in the research triangle. In other examples, such as ineparib and BIA 10-2474, poor characterization has led to patients being dosed with either inactive or actually harmful drugs. So over the past 10 years, there's been a real renaissance in our knowledge of how to develop small molecule chemical probes. So, one of my objectives today is to try to provide a public service announcement and highlight a couple of amazing resources that are useful for those of you who are developing or using them. One of these that I really like is the Chemical Probes portal. This is a website that offers expert assessments of chemical probes and can be a great resource to check out if, for instance, you're reviewing a paper that uses an inhibitor you've never heard of. Many researchers, including me, volunteer to keep this information current, and if you're interested in participating, I'm happy to get you involved. Another really great one is the Assay Guidance Manual, which is run by my colleagues at NCATS. This is an initiative that not only compiles information, but also offers seminars and workshops oriented around assay development and inhibitor validation techniques. So if you use or develop chemical probes to study chromatin, please check these out. I think it's an area where, the field can, be a leader and also make a highly positive impact. Okay, so next I'm going to tell you about the discovery and characterization of drug-like HAT inhibitors. Okay, so there we were, having done some housekeeping, ready to really set sail on our voyage of HAT inhibitor discovery. Working with NCATS, we devised an assay cascade where we first screened about 50,000 compounds for HAT inhibition by using a simple and robust CoA detection assay. We then triage our hits by moving them through our mobility shift assay. This is really where I have to tell, people in the audience, it's much easier sometimes to be a critic. Because what we found is that, to our dismay, most of the compounds that came out of these assays were fairly low potency and really littered with structural alerts, such as hydrophobic chains, oxidizable electrophiles, as well as oxidizable heterocycles. Now, fortunately, industry came to our rescue with AbbVie reporting in 2018 the medicinal chemistry optimization of a family of spirocyclic compounds with very potent P300 and CDP inhibitor activity. These molecules came out of a virtual screening effort against a structure of P300 bound to that high-affinity bisubstrate inhibitor lyse-CoA, followed by assay-guided optimization of a lead scaffold. Now, in addition to the really unique structures of these compounds, which allows them to bind to that challenging HAT active site, One of the things we found really incredible about this patent was the sheer scale of the effort, which involved the synthesis and evaluation of about 1,000 analogs in this single series. This is nearly an order of magnitude larger than an effort you'll usually see from a dedicated medicinal chemistry group in academia, let alone from a discovery chemical biology group such as their own. About a month later, the first characterization of these molecules followed in the literature via a Nature paper that detailed the activity of a small molecule HAT inhibitor called A485. This compound was found to potently inhibit P300 catalyzed H3K18 and K27 acetylation and exhibit anti-proliferative activity against a broad panel of cancers. Now, looking at the molecular details of this inhibition, it was found that A485 directly binds to that acetyltransferase active site in a site that overlaps with the acetyl-CoA cofactor, and biochemical studies also indicated an acetyl-CoA competitive mechanism. Now, in the immediate years since A485 was disclosed, AbbVie, as well as other industry groups, followed with additional high-affinity P300 inhibitors. These included IP300W, an A485 derivative that was optimized for in vivo stability, as well as CPI-1612, a completely orthogonal scaffold that was reported by Constellation Pharmaceuticals. Now, one caveat to these studies is that because of the timing of the release as well as their industrial origin, no one study had ever benchmarked these molecules against each other. There's also a little bit of a disconnect between the reported nanomolar biochemical potencies of these molecules and the concentrations required for cellular effects, which often were reported to be in the micromolar range. Now, this raised a question for our group, and possibly others in the field, which is that if you want to study P300 and CAP acetyltransferase activity, which chemical probe should you use and at which concentrations? To answer this, two really talented postbacks in the group, McKenna Crawford and Deepika Tripu, set out to comparatively analyze the properties of these drug-like HAT inhibitors. For biochemical analyses of these compounds, we worked with Steve Kales at NCATS, who used a time-resolved FRET assay to directly monitor P300's ability to acetylate a histone H3 peptide. Now, one thing we noticed in the literature was that the biochemical effects of these compounds had actually been analyzed using both different assays and also very different amounts of acetyl-CoA. This latter point is really crucial because if these molecules actually operate via an acetyl-CoA competitive mechanism, then we'd expect their IC50 to be actually directly proportional to the concentration of that acetyl-CoA cofactor. Considering this, we assayed each inhibitor at both a low and high concentration of acetyl-CoA. The latter, around 5 micromolar, is close to what's estimated to be the level of this metabolite inside cells. So when we compare the three compounds, what we find is that CPI-1612 down here at the bottom is by far the most potent P300 inhibitor, followed by IP300W and A485. When we perform the assay at high acetyl-CoA concentrations, what we find is that the IC50 shifts to lower potency, in each case, consistent with a competitive mechanism. Now, in addition to giving us some useful comparative data, one reason this fold change is so interesting is that acetyl-CoA levels also appear to play a role in dictating P300 inhibitor efficacy in cells, something that was really first shown by Mike Erb's group at Scripps, and that we've also done some work on. We can discuss this in the Q&A if you like. So co-crystal structures of P300 complex with that highly potent CPI-1612 molecule suggest some distinct interactions made by CPI that might be responsible for its stronger inhibition. Specifically, while both molecules bind to the acetyl-CoA binding pocket, CPI makes unique hydrogen bonds with backbone amides of a tyrosine and aspartate residue, and also forms a strong pi stacking interaction with tryptophan 1466 through its unique 4-cyanophenyl moiety. Neither of these are observed with, of course, with A485. So to test where the relative biochemical potencies of these molecules actually translate to cells, McKenna and Deepika analyzed their effects on histone acetylation. Here, just using MCF7 breast cancer cells as a model, what we find is that all three compounds catalyze P300 catalyze K18 and K27 acetylation, but not that of a control histone mark, acetylated H3K23. In these studies, we looked at all three compounds across an identical concentration gradient of 10 nanomolar at the low end to 5 micromolar at the high end. And this allows us again to observe that CPI-1612 appears to be the most potent of the molecules, consistent with those biochemical measurements. Postback Diamond Galmore in the lab collaborated with Ben Garcia at WashU to analyze CPI-1612 using quantitative mass spectrometry. And here again, what she found was that it causes selective loss of acetylation at K18, but not at neighboring residues, again, consistent with P300 selective inhibition. To better understand the capabilities of these three inhibitors across a wide range of cell lines, we submit them to the NCI 60 cell line screen, which is now headed up by Nate Cousins' group. What we find is that if we look at a subset of inhibition data I'm showing here, malignant cells really differ quite widely in their requirement for P300 CUP activity. A few cell lines like H460 or ACT116 happily tolerate P300 inhibitors, really no ill effects. However, other cell lines, particularly leukemia cell lines, are very susceptible. And for these cell lines, again, the potency of CPI 1612 exceeds that of the other agents. Now, one of the great things about the NCI60 is because so many compounds have been tested against it, scientists have streamlined methods to search all the data so you can actually compare how does a profile of different compounds cluster using an algorithm known as compare. Now, the hypothesis behind this is that if compounds share a common target or mechanism of action, then they should cluster together. Indeed, what we find is that when we use compare to analyze our P300 inhibitors, we find that of all 300,000 molecules that have ever been tested in this screen, they cluster most closely with each other, again, indicative of a shared or on-target mechanism. What's also, I think, pretty interesting is that they don't look all that similar to bromodomain inhibitor ligands. This suggests that even though HAT inhibitors and bromodomain inhibitors both serve to disrupt acetylation-dependent transcriptional signaling, their activities can actually be quite nuanced and distinct. Okay, so my last, 5 or 10 minutes now, I'm going to tell you how we're using chemical biology to facilitate new applications of HAT inhibitors. So in the first part of this talk, I kind of tried to answer one question you might have about targeting P300 and CUP, which is whether we could. Now that we have inhibitors in hand, we can start to ask a new question, which is whether you should. Now, the reason this is a good question, as some of you probably know, is that these compounds have the potential for on-target toxicity. So a substantial body of biological literature has demonstrated that P300 and CUP are actually essential for healthy development. One nice example of this came from my colleague Kai Guy's lab here at NIH, who showed that conditional loss of both P300 and CUP completely blocks the proliferation of mouse embryonic fibroblasts. This has actually led to the question being raised, for example, in the review shown here, as to whether the therapeutic window of P300 and CUP inhibitors will ever be large enough to actually achieve therapeutic effects. This led us to ask, how can we restrict the effects of P300 and CUP inhibitors? Now, one strategy to potentially accomplish this would be targeted release. For example, ADCs, photodynamic therapy, and targeted radionuclide therapy all work by targeting generally toxic payloads to a localized tumor context. To look at this possibility for HAT inhibitors, we analyzed first the crystal structure of P300 in complex with CPI 1612. Now, as I mentioned earlier, that secondary amine of this molecule makes a really critical hydrogen bond with serine 1400. This led us to predict that modifying this position with a stimuli-sensitive group, such as the photo cage highlighted in blue here, would actually limit the inhibitor's potency. To test this hypothesis, Yihang Zheng, a really talented synthetic chemist in the group, worked with Chris Leclerc's team at NCAS to produce and chirally purify several intermediates that he then assembled to produce a photo cage CPI 1612 analog. Now, biochemically, what we find is that when we modify CPI 1612 at this position, it reduces the molecule's inhibitor potency, as we expected, about 50-fold, shifting its IC50 from the nanomolar up into the micromolar range. McKenna Crawford optimized photo deprotection of the cage inhibitor. You can see on HPLC here, the cage compound slowly giving yield to the active molecule. And then, as a proof of concept, treated MCF7 cells with this compound, then irradiated them at 1 minute with high-intensity UV light. Now, this short time point, 1 minute, was chosen really to limit phototoxicity, and we don't expect it to drive anywhere near 100% release. However, really, because CPI 1612 is so potent, if we release even a small amount of this molecule, it's sufficient to drive light-dependent inhibition of cellular acetylation. This really establishes the principle that these inhibitors can be controlled via stimuli-sensitive release, and shows the importance of kind of understanding the potency and pharmacology of these compounds. Now, some of you remember it being suggested during the height of the coronavirus pandemic that intravital UV light might be a useful therapeutic approach. However, we really view this as a proof of concept. And we're most excited about taking this in the future is by using this attachment point to design derivatives of this molecule that have been targeted using more relevant therapeutic modalities, for example, as payloads and ADCs or using radiotherapy. Finally, I'll just end with a teaser for a second strategy that we're developing to restrict P300 CUP inhibitor toxicity, which is paralog-specific inhibition. This approach comes out of analysis of the cancer dependency map, wonderful resource I'm sure a lot of you are familiar with, which with which, among other data, has compiled the sensitivities of about 500 cancer cell lines to CRISPR-mediated knockout of about 15,000 genes. Now, when you look at the depth map data for P300 and CEP, what you find is that in many cell lines, sensitivity, which is indicated by blue here, is driven by loss of a single paralog, most often, if you really squint, P300. This is important because conditional knockout studies have actually found that in healthy adult animal models, loss of only P300 is well tolerated. This suggests that if you had a drug or inhibitor that inhibited only this paralog, it may be less toxic to normal tissues, but still effective against cancer. Now, the challenge again comes in differentiating these two enzymes. Shown here on the right is a crystal structure of the P300 hat domain, again, bound to CPI 1612. I've colored residues that are identical between P300 and CEP in white, and residues that are different in orange. Now, as you can hopefully appreciate, if you look at the small molecule binding site, proximal really to it, all of the residues are white. They're nearly 100% identical. This makes it really difficult to envision how you could build a molecule that can selectively inhibit either P300 or CEP via that active site. To solve this problem, we've turned to a strategy many of you are familiar with, known as targeted protein degradation. Here, what you do is you take a small molecule that binds to a protein of interest, such as P300, link it to another molecule that binds to ubiquitin E3 ligase. This creates a new bifunctional molecule that can then recruit the ubiquitinylation machinery to that protein, triggering its degradation. Now, really importantly, because targeted protein degradation requires the formation of these large ternary complexes with a protein of interest, it actually can be highly dependent on protein-protein interactions that are controlled and lie external to a small molecule binding site. This led us to wonder, could this approach also be used and enable paralog-specific inhibition of either P300 or CEP? To take a look at this, we benefit from a great collaboration with Rolf Swenson's lab, who runs a chemical synthesis core here at NIH, as well as a talented chemist in his group, Anver Basha-Sheikh, who helped us assemble a small panel of potential degrader compounds comprised really of 3 building blocks: that CPI 1612 hat inhibitor, a linker, and an E3 ligase recruiting moiety. Okay, so screening a small panel of these molecules by Western blot, we identified an optimized bifunctional degrader, which I also refer to as MC1. This is actually the first time we've disclosed the structure of this molecule, so maybe this is the first disclosure in fragile nucleosome history. Anyway, what we find so intriguing about this molecule is that it has a unique property of triggering almost a complete loss of P300 in HAP1 cells, while really leaving CDP largely unaffected. To keep it short and sweet, I'll just show you a few pieces of data around this compound. Mechanistic studies that we can carry out using a split nanoluke assay indicate that MC1, but not CPI 1612, the parent molecule, is capable of nucleating formation of a ternary complex between P300 and that VHL E3 ligase. We've carried out proteome-wide profiling studies, and when we assay the selectivity of MC1, what we find is that it preferentially degrades P300. This contrasts with another molecule, DCVP1, which is a really excellent dual P300 and CVP degrader that was developed by Christopher Ott's lab at MGH. Finally, in preliminary studies, what we find is if we assay MC1 against the NCI60 cell line panel, it appears to be associated with a unique inhibitory profile relative to that of the much more toxic dual P300 CVP degrader, DCVP1. Again, suggestive of the potential of this paralog-specific inhibition strategy to mitigate toxicity. Okay, I'm just running over, so I'll end with 2 take-homes. Today, I hope I've communicated the important role that chemical biology can play in developing small molecule inhibitors. We think that using new tools and assays to pressure test compounds at an early stage can really accelerate process, and in some cases avoid pitfalls that could lead to harm. Second, related to the intended title of this talk, I hope I've communicated that now, about 25 years after the discovery of HATs, histone acetyltransferase inhibitors have finally arrived. Much like HDAC inhibitors and bromodomain inhibitors that have probably been used by many of you listening, we think that small molecule HAT inhibitors, developed primarily in the last 2 to 3 years, can fundamentally change the way that we study this critical transcriptional process, and now actually start to open the door to new discoveries, as well as therapeutic strategies for targeted acetylation in cancer. Finally, I just thank my group of people who did the work. I tried to mention many different collaborators and colleagues along the way, but 3 fellows who really spearheaded a lot of the work around the CPI 1612 have been McKenna Crawford, Shuman Chen, and Ying Zhang. We're deeply indebted to, like I said, many collaborators, as well as NCI, for their generous support. Finally, thank you for your attention. Happy to answer any questions.
Jordan, thank you very much. That was really wonderful. And I guess it's great for all of us in attendance to be the first non-Meier lab members to see the structure of that molecule. Um, and so how do you, before anyone else enters something to Q&A, I'll start. Do you have any evidence or hypothesis for what is providing the specificity between P300 and CBP there? Like, is it preventing some interaction with like a chaperone or some other interacting partner?
Yeah, it's an interesting question. Like, I mean, so one of the hypotheses we had, let me go back a little bit, was that, perhaps it was, preferential ternary complex formation, right? What's interesting is we've ran this assay where we're looking at basically the ability to form the ternary complex using a split nanoluke assay, the equivalent assay with Krebs-BP, and we see it can also form the ternary complex. So that doesn't seem to be it. If you look at the biochemical data for CPI 1612, carried out with full length, I believe, protein from Constellation Pharmaceuticals, there is about a 3 to 5-fold increased affinity for P300 versus CBP. And it's possible that's just enough increased sort of residence time to allow, proper assembly of that ternary complex. We also have done some, AlphaFold multimer, predictions. You do see the potential for a few more interactions with P300 versus CBP. But again, it doesn't seem to be purely, it's not that, CBP can't form the ternary complex. It just seems to be less effective at triggering the full ubiquitinylation. But again, it's something we're still interested in trying to figure out.
So then I guess, since I've still got the floor here, actually, here we go. Dr. Harriet Kenyamu has raised her hand. Harriet, would you like to, let's see, I just allowed you to talk, so you can unmute and ask a question.
Hi, great talk. So I have a question on E3 ligases. So is there any preferential targeting of different E3 ligases between CBP and P300 such that whatever you link could allow the specificity?
Yeah, it's a great question. I mean, to my knowledge, it hasn't yet been, I mean, I very possibly could, have overlooked something, but it's not known, what E3 ligases endogenously, I guess, regulate P300 CBP. Again, we've done some, simulations basically, and it does appear that, P300 and CBP both prefer to interact with VHL over cereblon, which is another E3 ligase commonly recruited by PROTAC, targeted protein type modalities. And again, that would make sense because, kind of if you have a higher sort of baseline, I guess, interaction potential, maybe it's easier to push that over the threshold with a small molecule versus, something that would never interact, right? And so many people are doing this, but, we've tried to do this as well, where, if you have a target of interest, for example, can you, do in silico screening to look at, what E3 ligases might be most prone to bind to it, and then, try to recruit those ligases with the ever sort of expanding list of small molecule E3 ligase binders that industry and chemical biology are making. And so I do think VHL is going to be kind of preferential in terms of P300 CBP degradation. I mean, what's interesting though, I guess I should note, is that if you look at DCVP1, I didn't show the structure, but that comes out of Chris Ott's lab, that uses a bromodomain ligand, and they're recruiting in cereblon. And, again, just based on structure prediction, you wouldn't predict cereblon to really bind, to that portion of, P300, but I think we don't understand, and, modeling and prediction actually isn't that very good at, we don't understand how much plasticity is probably in these proteins and, can be sort of nucleated by a small molecule. So anyway, yeah.
Yeah, thanks.
All right. Well, yeah, so I'll continue where I was going before. I'm really curious about, P300 and CBP seem, they're the predominant functionaries when we think of like enhancer biology or transcriptional activation. But do you think that is there any crosstalk between their functionality or these inhibitors with other acetyltransferases, or are these like super specific to just these two hats?
Yeah, I mean, I think these hat inhibitors are pretty specific in terms of, I mean, we've done I feel like we've assayed them every different kind of way, basically. Now again, like, I mean, in terms of like crosstalk with other transcriptional programs and, you know, absolutely, right? I mean, I think that, obviously if you like look at depth map with P300, there's all sorts of different things it interacts with. So I guess I don't quite, sort of get at your question, but
Sure.
yeah, I mean, the inhibitors themselves are very specific, right? And I think,
Mhm.
I mean, I think the biology too that P300 and CVP carry out, like you said, relative to other hats, right? It is pretty unique, right? In terms of, I mean, I would say N of one, but I guess there's two of them. In terms of its capabilities, both, kind of the widespread number of non-histone targets they have, as well as just, the association with transcription factors, sequence-specific transcription, and enhancer function.
Right. Do you know from the tumors that have differential, or the cancer types that have differential susceptibility for P300 or CBP, do you observe any compensatory changes in the expression of the histone acetyltransferases, whether it's P300 or CBP or other hats?
Yeah, I mean, I think that's an excellent, question. I mean, one thing we've thought a lot about is, so our paralog specific degrader, one limitation of it, I would say, is that because we're targeting the hat domain, if we go to too high of a concentration, right, we can get dual inhibition, but we might just be degrading one, right? And, for example, if you went through the bromo domain, you wouldn't have that problem, right?
Right.
And so, but, the question I think that we're going to find out, and, to my knowledge, there are some groups working on paralog specific inhibition through the bromo domain. And, the question I think that we'll have an answer to is, are you going to get resistance? Are cells just going to upregulate CVP, right?
Sure.
Or, in our case, maybe we're actually going to be aided by the fact that at high concentrations, we're getting that on-target hat inhibition. And then, as, the molecule starts to, get diluted in circulation or whatever, could you still have, get a nice bump by getting that paralog specific, inhibition prior to, kind of losing all activity. And so again, I mean, these molecules are just arising, so people are going to be studying them a lot. And there's, it's a rapidly kind of moving field, I think, right now.
Absolutely. Well, it doesn't look like anyone else is putting anything in the Q&A, so, and we're hitting our time now as well. So I'd like to thank Kanish and Jordan both once again for another set of great talks here at Fragile Nucleosome. And as Aldous mentioned at the beginning, next week we will, or 2 weeks from now, we'll have a mini symposium for trainees. And you can check out the Fragile Nucleosome website on generegulation.org to look at those future events, or see us on Twitter to find the registration links. So again, thank you, Jordan and Kanish, and everyone have
Thanks.
a great rest of your week.